Hi everyone, welcome back. This is David Aguilar, COO of SHMN Inc. And I'm happy to chat today about our ABBIE technology and give a little explanation as to how it can affect you and our clients and other people that are interested in using what we're developing to help them and help their products and support their drug development needs. To understand ABBIE technology, you have to understand what gene editing is. And gene editing is changing genomic DNA, changing information. DNA is information. Programmers have the same idea, the same paradigm. They go into a code and they change the code. They're able to reprogram information in a way that allows for an output, like an application on your phone. Gene editors can do the same thing in a living cell, although not as efficiently as electrical engineers yet, but we are getting there. Gene editors today can change code to influence and help human disease. ABBIE technology is a gene editing tool. What we can do as a gene editing tool and a developer of a gene editing tool is create safe and efficient ways to reprogram DNA or information. And we use the ability of living information that's actually self-aware to facilitate our reprogramming. ABBIE is a processor. ABBIE is able to process large amounts of DNA in a specific way into a given cell, into a given genome. And we are leveraging this fully owned intellectual property that we've developed to make breakthroughs in science, breakthroughs in disease, breakthroughs into creating hope for people that don't have hope currently and are waiting for new therapies to help them survive and create quality life. ABBIE stands for A binding base integrating enzyme. That means our technology is able to bind to DNA specifically and integrate our payload, or you can think of it as the file. And we have the unique ability to incorporate large pieces of information or large files, which other technologies in our space are currently having problems doing. So what is ABBIE technology specifically as it relates to other gene editing technologies? Well, we fully own it. We have developed a technology that has many advantages over the other ones. For example, our technology does not induce what's called a double-stranded break. Now, what is a double-stranded break? Going back to the analogy of programmers, that's an error in the code, right? A double-stranded break is an error. Now, living code is self-aware, so it senses when it's hurt, and it can repair itself. Double-stranded breaks have actually been shown to be a pretty bad thing. They've been shown to what's called induce severe toxicities that weren't expected. ABBIE circumvents this by processing information directly into the cell of interest, directly into the DNA. ABBIE also has the ability to introduce very large payloads, or what you can think of as files. Competitors have this problem because they rely on the living cell, the living DNA, to repair itself. And this can be limiting because some cells are sick. Some cells just can't do that. And certainly, introducing larger, what could be considered a circuit, just isn't a natural thing for them to do. So our processor is very unique in that we can pull in large amounts of DNA, large amounts of information, without inducing a double-stranded break, and circumvent a lot of the problems that people are having in this space right now. This is the future of gene editing. Well, we introduce into our system a GPS signal. And that GPS signal goes directly to where we tell it to go in the genome. And we also introduce a file into the system that we want to incorporate into the genome. And of course, we're adding our ABBIE molecule as well, our enzyme, our ABBIE. In combination, this goes to a place on the genome. It incorporates our file or DNA of interest into the cell. ABBIE is a tool that scientists can use and cell engineers can use to engineer DNA of the cells of interest. Now, these scientists and engineers will be most likely addressing questions that will lead to drug development. So we are providing scientists tools and the resources to do their job and to help create new therapies for us. Industries that have anything to do with working with life or living organisms. Specifically, we're focusing on industries that are developing new drugs and therapies to support human health. So we are impacting pharmaceutical industries, biotechnology industries, research at the university level, and the government. The future applications of ABBIE technology will be used to address human disease, and we will do so by supplying tools and engineering services for the development of therapies geared toward cancer-related illness. Additionally, we are developing tools that will address genetic disorders that specifically are impairing and afflicting individuals that are inflicted with congenital heart disease. My vision for the ABBIE technology is to continue to develop the technology to be as precise and as efficient as we possibly can get it. Where we are now with our development is our ABBIE 1.0. You can think of it as the iPhone 1. We have plans that are going years ahead into an ABBIE version 7, 8, 9, and 10.0. And we also intend to incorporate and integrate augmented or artificial intelligence into our platform technology for future versions of ABBIE, more improved versions of ABBIE, as well as to facilitate the analysis of the data that we're getting both at the bench and in the patients for a more precise drug development platform. We are going for the right drug and the right patient and the right outcome. That's the future of ABBIE. And this is in line with the future of gene editing.